Animal experimentation, also called animal testing, has been used throughout history for important scientific and medical discoveries. Treatments that we use today, such as antibiotics, insulin therapy, modern anesthesia, vaccines for whooping cough, and other diseases, have been discovered by the use of animal experimentation. While so many advances have been brought from animal testing, many people object to the idea and believe it is now inhumane to use animals as scientific studies. These experiments take away their freedom and often lead to serious injury and discomfort. Animal testing should be banned in terms of experimenting with cosmetics and medicine because there are other alternatives to discovering human benefits without the process of animal cruelty. Animals have been used repeatedly through the history of biomedical research. Early Greek physician scientists such as Aristotle and Erostratus performed experiments on living animals. Galen, a Greek physician who practiced in Rome and was a giant in the history of medicine, conducted animal experiments to advance the understanding of anatomy, physiology, pathology, and pharmacology. For most ancient Greeks, using live animals in experiments did not raise any relevant moral questions. The supposed likeliness of humans to their anthropomorphic deities granted them a higher ranking in the scala naturae, or the chain of being, a strict hierarchy where all living and non-living natural things were ranked according to their proximity to the divine. This view of humans as superior would later influence and underline the Judeo-Christian perspective of humans' dominion all over nature, as represented by texts by Augustine of Hippo and Thomas Aquinas, Christian theologians of the Middle Ages. In recent years, the practice of using animals for biomedical research has come under severe criticism by animal protection and animal rights groups. Laws have been passed in several countries to make the practice more humane. Debates on the ethics of animal testing have been a thing since the 17th century. Animal testing in the United States is regulated by the Federal Animal Welfare Act, AWA, passed in 1966 and amended in 1970, 1976, and 1985. The AWA excludes birds, rats, and mice bred for research, cold-blooded animals, and farm animals used for food and other purposes. Toxicity tests to experiment the safety of animals and products were developed in the early 20th century. These included experiments that subjectively measure the irritation of chemicals in the eyes or on the skin of rabbits, and lethal doses experiments that kills half of the animals forced to consume, breathe, or be injected with that chemical. These animal testing methods are, unfortunately, still being used today. Many scientists continue to rely on animal testing because it is what tradition and current laws believe. Toxicologists often mention feeling more comfortable basing their judgments on methods with historical context and data than on data from new and emerging methods. However, this fails to show that even if it works on animals, it doesn't always work on humans. As a consequence, the lives of millions of our fellow creatures continue to be sacrificed. In 2016, the U.S. Department of Agriculture reported that 820,812 animals were used in experiments as USDA-registered facilities in the United States. This number does not include birds, fish, mice, rats, and reptiles, all of which are the majority of animals used in scientific studies. Of the animals reported by the USDA, the most commonly used is the guinea pig, which has been used for experimentation since the late 1800s. Guinea pigs, rabbits, and hamsters account for half of the animals reported by the USDA. The others reported test subjects were non-human primates, dogs, pigs, cats, sheep, and other animals protected by the AWA. Unlike field research, which involves observing animals in their natural habitats, animal experimentation takes place in laboratories in universities, medical schools, government facilities, and commercial facilities, such as those run by pharmaceutical and cosmetic manufacturers. The Dre's eye test, used by cosmetic companies to evaluate irritation caused by shampoos and other products, involves rabbits being forced in stocks by their eyelids held open by clips, sometimes for multiple days, so they cannot blink away the product being tested. The U.S. Department of Agriculture reported in 2016 that 71,370 animals suffered pain during experiments while being given no anesthesia for relief, including 1,272 non-human primates, 5,771 rabbits, 24,566 guinea pigs, and 33,280 hamsters. The anatomic, metabolic, and cellular difference between animals and peoples make animals poor models for human beings. Paul Furlong, professor of clinical neuroimaging at Aswan University, states that it's very hard to create an animal model that even equates closely to what we're trying to achieve in a human. Thomas Harding, professor at Johns Hopkins University, argues for alternatives to testing animals because we are not 70 kilograms of rats. Animals are inferior to humans and very different from them, so results from animals cannot be applied to humans. Over 100 stroke drugs that were effective when tested on animals have failed in humans, and over 85 HIV vaccines failed in humans after working well in non-human primates. Even when animal experimentations tests pass and are successful, they can still end up being harmful to humans. In the 1850s, a sleeping pill, thalidomide, was tested on animals prior to its commercial release. It is found to act as an effective tranquilizer and painkiller and was proclaimed a wonder drug for insomnia, coughs, colds, and headaches. It was found to treat morning sickness, so thousands of pregnant women take the drug to relieve the symptoms. More than 10,000 children in 46 countries were born with malformations or missing limbs. The drug was withdrawn in 1961 and 1968 after a long campaign. In 1937, a company created sulfamilamide using diethylene glycol as a solvent and called the preparation elixir sulfamide. DEG was poisonous to humans, and the company's chief pharmacist and chemist did not notice. He simply added raspberry flavoring to the sulfa drug, which he had dissolved in DEG, and the company marketed the product. The preparation led to mass poisoning, causing the death of more than 100 people. Despite many arguments on the topic of animal cruelty and experimentation, some positives include how drastically it has improved our knowledge of medicine and given a variety of cures. The California Biomedical Research Association states that nearly every medical breakthrough in the last 100 years have resulted directly from research using animals. Experiments in which dogs had their pancreases removed led directly to the discovery of insulin, critical to saving the lives of diabetics. The polio vaccine, tested on animals, reduced the global occurrence of the disease from 350,000 cases in 1988 to 27 cases in 2016. Animal research has also contributed to major advances in understanding and treating conditions such as breast cancer, brain injury, childhood leukemia, and many others. If thalidomide had been properly tested on pregnant animals, its potential for causing severe birth defects would have been discovered before the drug became legal for human use. Though animal experimentation helped scientists and researchers to discover many treatments and benefits for humans, there are also other more humane, cheaper alternatives to researching medicine and cosmetics. In glass testing, studying cell cultures in a petri dish can produce more relevant results than animal testing because human cells can be used. Microdosing, the administering of doses too small to cause adverse reactions, can be used in human volunteers whose blood is then analyzed. Artificial human skin, such as commercially available products Epiderm and Thinsert, is made of sheets of human skin cells grown in test tubes or plastic wells and can produce more useful results than testing chemicals on animal skin. Microfluid chips, organs on a chip, which are lined with human cells and recreate the function of human organs, are in advanced stages of development. Computer models, such as virtual reconstructions of human molecular structures, can predict the toxicity of substances without invasive experiments on animals. Because of today's technological advances, we are able to discover many more treatments and benefits without using animals. In conclusion, animal testing should be eliminated because it violates animal rights, it causes pain and suffering to the animals being experimented, and other alternatives for testing and research are available. In the words of PETA, the world doesn't need another eyeliner, hand soap, food ingredient, drug for erectile dysfunction, or pesticide so badly that it should come to the expense of animals' lives. Animals should be treated with respect and dignity. After all, humans are animals as well.